The enzyme has a heme component that has an iron center that is designed to attract oxygen. And then it's like a pitcher and a catcher. When the oxygen gets here, poof. These are the keys to air-based life on the planet.
Oh, I'm seeing it.
And this is the muscle fiber. And this is a close-up of one of those N-terminals. So there is more mitochondria granules for neurotransmitters. And for some reason, all the emphasis being on the neurotransmission and not on all of this metabolic process. We tested them 1 day later, 1 week later, 2 weeks later, 4 weeks later. There are changes that are happening. This device is already FDA approved for that condition and is the one and only device to stop and prevent neurodegeneration. Nothing else.
Hello, you're listening to Beyond Healthspan, a podcast that strives to decode longevity research to live better, longer, and without the guesswork. We focus on more than just aging, as we'll explore some fascinating science shaping the future of human health, some advanced strategies and breakthroughs that impact quality of life as well as holistic well-being. We are joined today by a truly distinguished guest and our kind of tickled fancy, Dr. Francisco Gonzalez-Lima. I'm a professor of neuroscience and pharmacology and toxicology at the University of Texas at Austin. Dr. Gonzalez-Lima, thank you so much for taking the time to share your insights with us and our listeners today.
Thank you for the invitation.
As you might have guessed, we are pretty big fans of yours, and I think your scientific body of work, the pharmacological approaches, even some of the lifestyle core competencies that you have published and shared on various other podcasts, I think are right in line with everything that we do here at Healthspan and what Rich and I believe in too. So pretty wide span of research that you have, brain energy metabolism, mitochondrial function, neurocognitive health, obviously aging, longevity stuff is what the Healthspan team does here. But you built much of your work around really understanding how the brain uses this oxygen and uses this energy. So I guess if you could share, and we'll start from the beginning, what originally sparked that curiosity in mitochondrial research?
When I was doing memory research in terms of neurobiology, this was the beginning of functional mapping techniques. It was, for example, the development of this glucose-based autoradiography, originally called 2-deoxyglucose, and then it came the fluorodeoxyglucose, or FDG, and I was with the group that developed this FDG autoradiography in animal models. So we use that as a functional neuroimaging tool in animals, and soon enough the approach was translated. But one of the issues that I have with things like glucose consumption, and then later on, with other techniques like functional MRI, is that they were all based on activity that was ongoing. So like a transient change. And they all rely, for example, in hemodynamic responses. I wanted, for the purpose of studying memory, something that will be longer lasting. This brought me to cytochrome oxidase histochemistry, which was developed as a qualitative technique by Dr. Margaret Riley, and I'm talking back to the 80s here. So I decided using that approach, maybe a way to do a metabolic marker that I could use for mapping where in the brain there were changes in what I refer to as metabolic capacity. In other words, not what is going on now, but what you see in the brain after learning takes place and memory is formed. How is the metabolic capacity of the brain changed by these learning experiences? So that's how I started to use these techniques to do this kind of mapping. It was not as an interventional approach, but trying to see how can I benefit from that. And then I had to develop in the early 90s a quantitative enzyme histochemistry method to be able to do that with the more subtle changes that happened during learning and memory. And that was the first quantitative enzyme histochemical method. The beauty about that was you could do the experiments in vivo and then look at the changes that stay there. That's how it all started. And since then, I have been doing a lot of work around cytochrome oxidase.
I'm glad you brought up the FDG PET. I was hoping you would, and I think Rich and I almost owed you a thank you because we actually used that in our stage 4 metastatic breast cancer trials while we were implementing the ketogenic diet back in grad school, and it was really fascinating to see that glucose uptake. I thank you for that, for being part of that.
Yes.
Much appreciated. Some of the things that came up just based on what you were saying there that were super fascinating within a line of research or a research career, research arc, if you might call it. It's not always a linear path. And you started kind of working on the imaging side, then you got into more of the histochemistry or the immunohistochemistry, and it wasn't interventional, but some of your more recent stuff does seem to be more interventional. Looking back at your career, like what moved you out of imaging? I know it had something to do with like the timing, and wanting to look at things in a little bit longer duration. And where has your research trajectory changed, I guess, or your career focus moved?
Looking back, I would say one of the things that impressed me was the importance of focusing on energy metabolism versus, for example, neurotransmitters or neurotransmission. The whole field of neuroscience was really influenced by neurotransmission. There was a big neglect on energy metabolism and how the brain was dependent on that. Through my work with this animal model, it was very clear. I mean, the brain doesn't have much in terms of energy reserves, and energy metabolism seems to be an ongoing process that needs to be maintained. And as soon as you ignore that, real deficits start happening, and from very subtle changes to atrophy and neurodegeneration. What in those days I like to refer to it as metabolic pharmacology, trying to see whether we could address this. And this was solidified when I first used our quantitated cytochrome oxidase histochemistry to map the postmortem fresh brains of Alzheimer's patients. We wanted to know if energy metabolism was taking an important role here because all of the non-invasive techniques done in vivo suggested this. For example, you mentioned the PET, FDG PET. It was very clear from FDG PET that there were problems with glucose uptake in these patients, not only when they had Alzheimer's disease, but even individuals that carry risk factors like these ApoE4 alleles in their 20s. They already had hypometabolism as reflected by FDG uptake or cerebral blood flow measures not done non-invasively in vivo. I tried it with brain banks throughout the US and elsewhere, but the tissue was too old, not of sufficient quality to do any of this work. Luckily, there is a place called Sun City where retired communities live. And in Sun City, Arizona, they have a center that takes care of them. And lots of these people are generous and donate their brains for research. So one of my former trainees, John Bala, and I essentially went there, had a deal with this Health Research Institute, and we will take the brains from people as soon as they will pass away, take it out, and I will start dissecting the brain quickly and freeze. And the deal was I will get a piece for me and another piece for the institute. And importantly, they have lots of fresh frozen material that then I was able also to work with. First, we found all of these control brains, that is, individuals that were old, but didn't have any evidence of dementia or Alzheimer's brain, and then sort of match them to individuals that did. We spent the whole summer doing this and send all the tissue, frozen tissue, back to Austin. I share a room. We were roommates with my graduate student. I think we cannot do that anymore because we were working, any time of the day, the pathologist will tell somebody passed away. He will certify the test and we'll be there. I will provide to them my expertise in neuroanatomy to dissect out all of the little pieces. That's how it started. But the important thing was the outcome of that study, because it showed that cytochrome oxidase was really a key player in the changes that were happening in those brains. And at the same time, it showed these areas that were more hypometabolic, they have no relationship, for example, with the accumulation of amyloid deposits. Of course, regions in which there were these neurofibrillary tangles, by the time you have this tangle inside these neurons that you can see, those neurons are functionally or metabolically dead. They're just like ghosts. The deficit there can be easily ascribed to a consequence that happened. The neurons are no longer viable, and therefore you can have a hypometabolism. but not in these other regions. And that's where we found that, sure enough, matching the in vivo data, especially FTG and cerebral blood flow, the posterior cingulate cortex is where the largest deficit in cytochrome oxidase activity was found. This created this link, and we had internal controls, like, for example, looking at the primary motor cortex on the same brains and not seeing these levels, looking at age match and well-matched controls and not seeing these changes, also doing laminar analysis and determining that the changes were in the superficial layers where you had the rich neuropil, all those synaptic contacts that are high energy demanding and require more of this oxygen consumption and ATP production to sustain this synaptic activity. So there were many lines of evidence suggesting to us, including that the progression of the disease, the longer somebody had been demented, the larger these hypometabolic cytochrome oxidase deficits were.
It's amazing to me that just based on the brain, right, being such a small organ relative to body weight, 20% of your total energy expenditure goes to the brain. So obviously high energy demand. So if there's an energy issue there, that would manifest in probably some poor outcomes regarding like actual, brain health or disease rates associated with neurodegenerative diseases and things like that. So it's crazy that there wasn't a ton of research there, but I'm grateful that you saw it and then you hopped on it, because that's probably a big key to like some of the amazing stuff that you've been able to, follow up with.
And because our work was in this neurobiology of learning and memory, the first thing that I did was in animal models inhibits cytochrome oxidase function. You cannot do this to a large degree. Animals cannot survive without being able to use oxygen. But if you were to go all the way to, let's say, 30%, the first thing that will be impaired will be these memory processes. They require coordinated action from many different areas of the brain. And of course, this limbic cortex, like the cingulate cortex, are part of these circuitries. In other words, we tested this experimentally. Can we see changes that lead us to believe that the memory disturbance is a primary initial change if you compromise cytochrome oxidase activity? Or if we will, with a cannula, put it right there in the posterior cingulate cortex in an animal, we will create a model that resembles like a mild cognitive impairment situation in the animal, and all the other functions will be spared after that more localized metabolic inhibition.
I have more questions, but I want to make sure I'm not leaving Brandon out.
I trust that we have very similar questions. So.
A slight detour for folks with regard to like the cytochrome C oxidase activity. Would you be able to give like a brief refresher regarding like complexes 1 through 4, mitochondrial function? How is it that cytochrome C oxidase ends up being like this pivotal or prominent point where, that's where if you have energy issues, and maybe it's dispersed more than that, but it does seem like that is a very important focal point. What is it about complex 4 makes a difference? Can you place it for folks? And then we'll get into the interventions, and then hopefully that'll bring everything together for folks that are listening or following along.
Cells like neurons or muscle cells, they are highly dependent on oxygen in order to be able to generate energy. That whole process is called oxidative phosphorylation. Oxygen is the entry point that allows phosphorylation of adenosine diphosphate and adenosine triphosphate, or the primary substrate. And essentially, what the machinery, biochemically speaking, that was created is a machinery that you have a phosphate bound, that when you break that phosphate away is a so-called exothermic reaction that it provides calories. And it's, in fact, the term calorie comes from that heating that happens. And if you remember in chemistry, you could have some reactants in a beaker there sitting and nothing happened. And then you have your burner heat up the solution, and then these reactants became a product. All of the ones that we refer to as energy metabolism that utilize that breakage of ATP are reactants that have to be synthesized, but then nothing happens until you provide the energy to the system, and then you get the products that you want. And by doing this, you can control this, and you have catalysts that facilitate that pass from the reactants to the products, even though they are not part of the ingredients that are the products or the reactants. They facilitate that. And that's what cytochrome oxidase is. And the particular reaction is the one in which the oxygen is used. And that oxygen is catalyzed into water. That wouldn't be that important if it's not that within the mitochondria, this reaction is being enclosed in a system that when the oxygen becomes water, it pumps some ions across these internal membranes of the mitochondria that are responsible for another enzyme adding this phosphate to the adenosine. So the so-called oxidative phosphorylation. But what is it that drives that process? Oxygen is the ultimate electron acceptor in nature. The process of removing electron from any chemical is called oxidation. What happens inside the cell is that the cell captures these other original organisms that were bacteria, so-called mitochondria, that were very efficient at doing this process of using oxygen in a way that was coupled with the production of ATP. But where do we get the electrons from? Well, normally we get them from the food that we eat. The nutrients, they provide also building blocks for doing synthesizing things. But from an energetic point of view, their role is to create so-called electron donors, like NADH being the most important one. These electron donors donate that to the so-called electron transport. So these large protein complexes inside mitochondria were specifically selected, designed for their ability to capture electrons and move them to oxygen. And the last movement, these so-called four complexes, 1, 2, 3, and 4, is complex 4 is cytochrome oxidase, the one that will pass that on to oxygen and that reaction. So it's said to be then the rate limiting reaction, the one that is coupled with the amount of oxygen that is consumed. By oxygen consumption, we're meaning that oxygen becomes metabolic water. So these are the keys to life in what we call aerobic life or air-based life on the planet. How can we use oxygen to provide this metabolic energy? And cytochrome oxidase is that key player there. The electrons can go in through complex 1 or complex 2. Then the electrons jump. There is a little small so-called electron carrier that is called cytochrome C. And that's the one that normally passes it from complex 3 to 4. Later on, you will see that it's important why they are called cytochrome from the point of view of how they absorb and reflect light. Any chemical will absorb some light wavelengths and will reflect others. But the key here is when these complexes do it, especially cytochrome C oxidase, these photons are pushing electrons out towards oxygen. They are doing the same that normally we get from these electron donors that come from the nutrients that we eat. So we can bypass the whole process, the electron transport chain, by directly applying light that is absorbed by that cytochrome oxidase and pushes electron. Is an electron photoelectric emission phenomenon first described in the 1930s by Albert Einstein. That was the most influential actually work that led to his Nobel Prize, but forgotten afterwards. It was believed it had only happened on the surface of some metals. But no, this phenomenon, especially these complexes that are specialized to move electrons out to oxygen, they are very sensitive so that photons can do that. And of course, this led me to the use of photobiomodulation. But before I went there, because of my training in pharmacology, I look for how can I get some other electron donors that can replace the regular NADH and FDH. And that's what led me to methylene blue.
I could probably listen to that last 5 minutes as like a bedtime story every night on repeat. That was beautiful.
Thank you.
I think we were exactly where both Rich and my brains were going with both the methylene blue and the NIR as interventions, as you called out a couple of minutes ago. So we're both very interested in both of those, and that's probably the appropriate place to go next. And then I would also love to hear, maybe as a prelude to that, how you would assess mitochondrial health, or what kind of tools you would use to say, hey, maybe I need to do NIR or the photobiomodulation or methylene blue or something like that as the precursor to intervening.
Yes. Let me start with that. So, for example, in biochemistry, one of the things we used, we were using methylene methylene blue. And methylene blue, low concentration, could become an electron cycler in the sense that at low concentrations, the amount of oxidized and reduced methylene blue will reach an equilibrium, and they will be taking electrons from the outside, the immediate environment, but also donating electrons to there. But the beauty of that, there are some compounds that can do some of these. It's a phenomenon that's referred to auto-oxidation. However, methylene blue was special because it was the first, not only the first synthetic chemical that was used for medicinal or pharmacological applications, but when it was injected to a rat, the discovery was that if you were to open that rat after that in vivo injection, you found that blue stain was captured primarily by the nervous tissue. So you could see the brain and all the nerves that were taken out. They didn't know at the time, this was about 120 years ago, why could this be? But really, the real reason was because these regions had large accumulations of mitochondria. In fact, this methylene blue staining was used by the founding father of modern neuroscience in the 1880s and 90s, Santiago Ramon y Cajal. He had used other techniques to identify, for example, these so-called synaptic ends that we call now dendritic spines, where synapses happen, where there is a lot of energy use, loss of mitochondria. Any place that you have a synapse, the presynaptic and postsynaptic ends are loaded with mitochondria, not just in the brain, but for example, in a neuromuscular junction. You can see it on both ends because you need all of this energy to support this chemical neurotransmission. Well, Cajal, some people say that these little spines were artifacts of his silver impregnation technique. So he redid the studies using methylene blue stain. And microscopically, he found that he could see all of these spots that were the dendritic spines because that's where the methylene blue was accumulating, where the mitochondria was. So in reality, methylene blue is mostly an mitochondrial stain because in this lower concentration, it has affinity for this electron transport. That's the place inside living cells where most electron transport and movement is going on. In order to maintain this cycling that happens when it's at equilibrium in the redox oxidized state, it gets into that environment and it starts cycling. So methylene blue feeds electrons to that electron transport. And in fact, if you have too much oxygen that is being delivered to the tissue and cytochrome oxidase cannot reduce all of that oxygen to water, that free oxygen there that is not fully reduced is the one that forms this reactive oxygen species. This is like superoxide. This is essentially an oxygen that wasn't fully reduced because there wasn't enough cytochrome oxidase there. Like when you're running in your, you can breathe more air, but you may not be able to keep running. And the limit is that cytochrome oxidase level in the tissue. Of course, you create an oxidative stress. You get more oxygen and more perfusion of blood than you actually need in terms of the oxygen that will be metabolized. So methylene blue does this double whammy that it can then directly bind to that oxygen to finish with the reduction. Methylene blue not only accelerates the electron transport to oxygen, but whatever oxygen is not fully reduced, it directly interacts with this. So I got two things that no other intervention has given us before. The ability to increase ATP through this oxidative phosphorylation and the ability to reduce oxidative stress to prevent oxidative damage. Because, for example, some people can accelerate oxygen consumption, but at the expense of oxidative stress that may produce damage. Or the other people do the opposite in trying to prevent the oxidative damage. They will have like caloric restriction. Yes, you lower your oxidative damage because you're not using as much oxygen, but you are not generating ATP energy. However, with methylene blue, you can have it both ways. You can accelerate ATP production and directly reduce oxygen at that very initial step of superoxide. Because what the mitochondria does afterwards, make it closer to water by making it into, for example, peroxide. You have this superoxide dismutase, and then you have enzymes that act on the peroxide that make it in a hydroxyl radical. So we're trying to make it more into water. All that cascade that happens. But in the meantime, these highly reactive oxygen species are binding to all the molecules and producing this oxidative damage that is interfered with whatever, for example, with the mitochondrial DNA that is not very protected. Mitochondrial DNA is circular DNA, like bacterial DNA, and doesn't have any of these other protective proteins in it. So it gets mutations very quickly from these reactive oxygen species. Methylene blue then decreases all of these processes, and you can see more energy produced and less oxidative damage.
I don't think that's ever been explained to me, and I spent a lot of time focused within some of my coursework on like neuromuscular junction and motor units and that, like all the physiology associated with that. No one that I can recall, I never remember learning that like these neuromuscular junctions are like full of mitochondria.
If you want, I can show you an schematic of electromicrogram of that neuromuscular junction.
Yeah.
Very quickly. Let me see.
Bear with us. This is awesome. Oh, I'm seeing it.
You see this, it's an axon that is coming here, and this is a muscle fiber. You can see some of these contractile elements here. And this is a close-up of one of those end terminals. Everything that you see here is all mitochondria. So there is more mitochondria than granules for neurotransmitters. It is completely loaded on the presynaptic and the postsynaptic end. And of course, this is happening as well in neuron in synaptic contact. And for some reason, all the emphasis being on the neurotransmission and not on all of these metabolic process that was needed to support that. I understand, one of my former professors, Dr. Jose del Castillo, he is the one who discovered quantum release together with his postdoctoral advisor. The phenomenon, again, the emphasis of neurophysiology was all in neurotransmission processes.
That comes full circle and even makes sense with like, you chose to focus on metabolism, but I could see why if you're like following, the science, the data, the evidence, and then like the imaging information that you had access to, that makes a ton of sense. It doesn't make a ton of sense based on like the textbooks, I guess, that I've seen, where I don't know that I recall seeing like nothing like that in terms of density of mitochondria. You see the dendritic fingers and the synapse, and like you got the ACH, the acetylcholine vesicles being sent back and forth, but you don't see a lot of mitochondria in those images.
They point out what they want you to see.
Okay. We need to get you in the textbook game, Dr. Gonzalez-Lima.
Yeah, I have invitation, but as long as I can contribute original research, I'm going to be doing that. Next month, I will turn 70 years old. I will continue as long as I can get original data on this, and especially use this in clinical applications, start using that. But just briefly, I can tell you that I was intrigued by the red to infrared light, again, by the work of Margaret von Riley. This is one of my scientific heroes. She's retired now. She demonstrated in ganglion cells from the retina that in those cell cultures, you could prevent mitochondrial toxins from affecting those ganglion cells that are really neurons. One of my trainees, Julio Rojas, was very intrigued by this process. And then I proposed to him, let's study this as your dissertation work. He was brilliant. And so we moved from these cell cultures. First, we did some in vitro work to show that in brain homogenates, for example, or synaptosomes, that we could see the same kind of phenomena. And then we move to the eye, passing the light directly to the eye in animals, different models that we have in mice and rats. Why in the eye? Because I was sure then that the light will get to the retina. And then we have mitochondrial toxins that will produce neurodegeneration there. And we could demonstrate that with essentially what it was at the beginning was red light, doesn't have any problem getting through. You could prevent retinal ganglion layers from atrophying and degenerating out. And you could demonstrate in vivo in the animals that their visual acuity was spared. You could do all of these tests. This was his dissertation work. And in the process of doing that, then we got encouraged and tried to do it transcranially. And sure enough, we then used the cytochrome oxidase quantitative histochemistry, saw the changes in the brain tissue. And we published this in 2008 for the first time in the Journal of Neuroscience. And it was a first demonstration in animal in vivo that you could do this in the retina and also transcranially, and that this will affect cytochrome oxidase activity, which was our target. This is how it all started. Now Julio is he's an MD, PhD, and now he's a professor of neurology at UC San Francisco. Many other people also contributed. So we moved from there, did some other animal work, but it was not until 2013 that we actually produced the first study with photobiomodulation that was a control study showing that we could improve cognitive functions in humans by stimulating the prefrontal cortex. So by giving resources to these, you affect many of these we call prefrontal-based cognitive functions. But why was important to use the light? How come it did this to cytochrome oxidase? Cytochrome oxidase happens to be the major intracellular acceptor of photons in that range of red to near infrared. In other words, light could be absorbed by other chemicals, but the main one inside cells that will absorb this range, what we call optical window. And this is important too, because an optical window that the photons can get in, the ones that can get into the brain, then are going to be primarily absorbed by cytochrome oxidase. And when that happens, essentially those photons, we're going to push electrons out to oxygen, like I mentioned before, and the cytochrome oxidase becomes oxidized. So it is a process that I refer to as photooxidation. Many of the people working in photobiomodulation, they only refer in more general terms, or it acts on cytochrome oxidase, it upregulates, it facilitates activity, but they have not narrowed down what is happening is that you're doing a photooxidation process. And of course, the preferred sector of this for which the enzyme is specialized is oxygen. The enzyme has a heme component that has an iron center that is designed to attract oxygen to it, to its catalytic pocket. And that oxygen comes from this other compound, hemoglobin, that also have that heme pocket to transport the oxygen. And then it's like a pitcher and a catcher where oxygen goes from one to the other. When the oxygen gets here, poof, it closes this and allows the electrons to be taken up by oxygen and water to be produced. So it is a fundamental process of life on the planet. We call it high energy phosphate, one that when broken down releases this calorie. And from there on, then we started testing in different dissertations, sustained attention, different cognitive functions in healthy young adults, working memory, executive functions, rule-based learning, all of these things that we knew prefrontal metabolism, by benefiting that, we could potentially influence them. Our next step was then, how can we use this in clinical population, which is what we have been doing now. So in clinical populations, I had several characteristics that I wanted, but the most important one was that there will be evidence of mitochondrial dysfunction in that particular clinical condition. And two, that the prefrontal cortex, which we could target through the forehead, was a key component of that particular clinical problem. That way, we will increase our chances of increasing energy metabolism there, having an impact. Going through the hair is not a good idea because the hair has melanin and it absorbs a lot of light. That's not a problem here. And even if you have a very pigmented skin, it's not a problem because the deeper layers of the skin are the ones that contain the melanin. If we have a white skin, we still have that layer of melanocytes deeper in our skin. So maybe a discovery for some people in your audience that we are all black, a little deep in our skin. We apparently come from a descendant that was black. We have now published some clinical applications with depression, 2 different studies. The latest one was, I think, the best control one. We published in bipolar disorder, but there it was not trying to take care of the depressive emotional changes, but it was trying to improve the cognitive performance of people with bipolar disorder. You can do mood stabilization fairly well. However, nothing pharmacologically is being done to prevent this accelerated cognitive decline that happens in the individuals with bipolar disorder, and many times more than people of the same age. By doing this in the prefrontal cortex, we were able to show we can improve their cognitive performance, even if they are remitted, that they are ready. In the course of doing these studies, we found out also that the best way to use the photobiomodulation transcranially was synergistically with another intervention that will engage that cognitive function. In other words, you create the cognitive demand by, for example, doing cognitive behavioral therapy. It requires you to do cognitive processing. If you are very depressed, you cannot think because the prefrontal cortex can be in the wrong direction. For example, when you have this rumination of negative thoughts and negative emotions, and you don't want to feed that. In our hands, if somebody is ruminating these negative thoughts and you feed the prefrontal cortex, it goes in the opposite direction, like looking at the glass and saying, oh, it is half empty, not half full. That's what somebody with depression will do. They will always zero in negative aspect or looking at the sad faces or sad words as opposed to neutral ones. So you want to get then the psychotherapy to move out of this. Once you start responding to the psychotherapy, then you fit them with the photobiomodulation and you facilitate that process by providing energy to the system. And we've done that now with depression and with bipolar, and have ongoing studies with ADHD, attention deficit hyperactivity disorder, because again, it matches those criteria: prefrontal involvement, some mitochondrial dysfunction, and that we know in people that don't have the disorder that we can improve this particular sustained attention. And we're finding beautiful results with reduction of impulsivity. That is the main reason that your attention goes away. And those are ongoing. That will be the dissertation of one of my talented students. We have more bipolar work to do. Now we want to do with people that have in different phases of the mood changes. The other major study that we are just starting out because of these delays that I mentioned before is autism spectrum disorder. And we're doing that across the age spectrum, from adult to adolescents to children, because this is a neurodevelopmental condition that persists in most people throughout life. And we've done some preliminary work with cognitive aging, older population, all the way to 90 years old, MCI, subjective memory complaint, and we are planning a study that will address that group. But in that study, I want to use a combined strategy targeting mitochondrial energy metabolism, which aligns with your interests. In other words, I will have the pharmacological arm that will have the methylene blue for its electron cycling capacity. It will reach all of the brain tissue. I will have the photobiomodulation to this prefrontal region that we can target that for especially cognitive processing and emotional regulation. And then the ketogenesis. The ketogenesis to facilitate an alternate route as we grow older, our ability to use glucose, to uptake the glucose so that we can do these electron donors or the electron transport is compromised. We provide that alternate metabolic route that is more preserved as we grow older, and that you can actually train by having some periodic caloric restriction, facilitate the upregulate the enzymes that will shift towards that pathway. And so one can combine these things in a way to optimize, and that's what we want to do next. But when you combine things scientifically, you get a lot of resistance. They want you to test one thing at a time. Well, we have tested the methylene blue, and this is my next challenge to get that approved so that we could start with that in older population, especially with mild cognitive impairment. However, I have not done Alzheimer's disease because oftentimes by the time these people get diagnosed, there is a lot of degeneration that has happened. I can preserve what is still viable, but I cannot reverse any of that atrophy. And I am very mindful of this, not to mislead anyone on that. And to tell you the truth, the people in the amyloid dominate that field, and they won't allow any other interventions. You cannot get any funding anywhere. If there is one of these persons there that is amyloid religious fanatic, then there's just no possibility they would not allow it. But that's where we're headed. We still have lots of work to do. None of that will work well in the older brains if we cannot improve the vascular supply. We need brain vascular perfusion. The role of oxygen supply is vital because we can get the chemical machinery upregulated, but if we cannot get the oxygen there, we won't be able to get oxidative phosphorylation. So this is a limiting factor. And we have tried, measuring, for example, carotid ultrasound as measure of blood flow to the brain. And it's very tight with age. Actually, it's completely collinear. It doesn't provide to us any additional information than your age. The older we get, the less perfusion we have, at least through the carotid, to the brain. The photobiomodulation helps a little bit on this because in the process of the photooxidation of cytochrome oxidase that allows oxygen to enter the catalytic pocket, it releases nitric oxide, another gas that is very similar to oxygen, that also gets into that pocket. And it's normally used as what's called an allostatic inhibitor. When you have too little oxygen in the tissue, then cytochrome oxidase itself becomes a nitric oxide synthetase. In other words, it takes nitrates and makes the product of nitric oxide, that gas, which produces a local vasodilation in the area where the gas is being produced. More than perfusion happens in the tissue, more oxygen level, and then the enzyme switches to its preferred substrate, that is oxygen, and continues the process of oxidative phosphorylation. We continuously have this as a regulatory negative feedback mechanism. The photobiomodulation does help in those two, releasing the one that is already in the pocket and letting the enzyme, when there are low levels, particularly in older people, to produce more local nitric oxide where you need it, not just anywhere else. It's where the light that you are acting on. So these things are helpful, and we'll see how we can continue with that work.
That's amazing. And I appreciate, because you kind of gave like the tour of your research, I guess. We were going to ask you the question because obviously your 2013 publication where you focused on the kind of synergism between the ketogenic or the beta hydroxybutyrate, the ketogenic diet, plus the methylene blue, plus the near infrared, the laser, therapy and things like that are more targeted, like the combination of the three, as you had mentioned. Crazy. I think you were way ahead of the time, anyone else at that point when you published that. Obviously, it's 10 years plus. And then we saw like 2015, there was a similar study, but it was more focused on methylene blue with the near infrared, but there wasn't as much on the focus on the ketone. So what Brandon and I were talking before in our prep, like what's been going on with Dr. Gonzalez-Lima's research in terms of like, was there something with ketones that was either unfavorable or funding or IRB related, or same thing with methylene blue, like, because your more recent research in the last 5 years or so has been very focused on more of the near infrared and the laser.
Yeah.
And I was like, well, that's, maybe there's something there, the pharmacological stuff or the synthetic stuff, or it's hard to get people to adhere to like a ketogenic diet in some cases.
No. Two things. One, I follow a systematic approach, methylene blue, photobiomodulation. Now is the time for the ketogenesis. And number two is that I saw that other group that you have been part of successfully doing that work in parallel. And I was so happy to see that going, and so that I could resolve some of these other questions with these two approaches. And right now, in the clinical world, people are using these combinations in order to satisfy our scientific rigor. We need to do a sham controlled, randomized study. And I want to at least use those three elements. There's no scientific reason not to combine this. Absolutely. Especially the ketogenesis and alternate route that is not being impacted by these other manipulations. And I'm all for it. It is something that luckily has been preserved as we grow older, especially in that population. Is a key to try. However, through changing your diet is very difficult. people do not tolerate well these changes. So having other ways like adding a supplement, whether it's the medium chain triglycerides or ketones themselves, like ketone esters, that seems to me the more productive approach in these days. There is a lot of evidence that you can have these ketogenic diets, but for example, with epilepsy, even bipolar disorder with benefits, but it's very difficult compliance wise. I think it's possible to have the three things, combine them, and see how they benefit various clinical or just regular healthy individuals, especially as they mature and grow older. I personally have tried the three of them, and whenever I am challenged, I take advantage of them. There is no reason not to do that, in my opinion.
Just because I know we're probably going to get asked this, just as like the senior health coaches here at Healthspan, and for our listeners who are going to want to know, is this something that maybe for like the more longevity focused person who's not dealing with multiple sclerosis or Alzheimer's or things like that, is something like NIR something that you would recommend the same way you would say you should resistance train 3 days a week? Is it something like you should do cyclically or only if symptoms arise, or if you feel like your mitochondria might be damaged a little bit, or what would you kind of recommend for the average bear?
Yes, I definitely think at least once a week. It lasts. One study that we submitted for publication, it hasn't been reviewed yet, give us an idea of how long the changes happen after one session. And you have changes that happen throughout time. For example, in the animal, the first day, these prefrontal regions are the ones that are showing the main benefit with cytochrome oxidase. As we tested then 1 week, 1 day later, 1 week later, 2 weeks later, 4 weeks later, and even after 4 weeks, there are changes that are happening. And some of the changes are not intuitive at the beginning because, for example, if you facilitate some of these prefrontal functions, like this medial prefrontal area, the way the brain is a network, some of the areas are going to have to go down in their energy utilization. To give you one example that people are very familiar now, you facilitate these medial prefrontal areas with the photobiomodulation, and you reduce amygdala activity, which is something that many people have been trying to accomplish. Well, because this medial prefrontal acts in some way as a brake at the top button inhibitory control of that emotional related limbic area. Throughout the 4 weeks, you have the cytochrome oxidase upregulated in the prefrontal cortex, and by 4 weeks, the effects are enough that it is down-regulated the amygdala activity. And so these are things that potentially could have also impact on emotional regulation. And we have tried this in people with anxiety disorders, we call pathological fear. Photobiomodulation alone can significantly reduce this pathological fear, a whole array of anxiety disorders. We have published that already. It is clear that it's not just facilitating energy metabolism in that target, but what that target is doing with other parts of the brain as part of this network, especially in somebody that needs that inhibitory cortical descending influence to come down. Once a week has worked for many things that we don't, but there is a potential if you use LEDs, that we're starting to do this with the autism spectrum disorder for safety reasons. I didn't want children to be exposed to lasers. So for that population, we have collaborated with engineers and LED devices. They're similar to our lasers, not the inexpensive ones that you can find all over the place. These are the same wavelength, and they have power levels comparable to what we have, but they don't have these safety concerns. And those you could potentially take home and use every day, especially in a population that is a clinical population. In the autism, because there is a clear interaction between the brain and the gut with the so-called microbiota, we're also stimulating abdominally. We are pioneering that abdominal photobiomodulation. We don't only this in pilot studies, and we have incorporated this now into our large study, 280 participants. So we're going to be analyzing the microbiota from stool samples to look at the impact of this as well.
You've more than convinced me. Not that you need to convince me of anything. I'm a big fan now of NIR. I don't have any devices. I don't know where to go to get a device that would meet the Dr. Gonzalez-Lima stamp of approval for potency and such. I'm looking at your 2025 study right now where there's a visual provided.
That thing that you see
Yeah.
there is not for stimulation. This is to measure oxygenated hemoglobin.
Gotcha. Okay.
To demonstrate intervention is actually improving the oxygenation to this prefrontal tissue.
Gotcha. Okay.
Two of my former trainees, one that I was a co-advisor, he did his dissertation on these lasers and help with many of these devices at my lab. He has a small company here in Austin. And now one of my current graduate students who has an engineering background is also trying to develop his own small company. So far, what I can do is just recommend the ones that we have tested in our lab for research purposes.
Not going to be the standard red light that you can like grab on Amazon. This is something much different,
Fortunately.
Okay, that's fine.
Unfortunately.
That's fine. People are going to want to know, like, where do I get this?
Right now, there's one source, and soon there will be another. I am not financially linked to these sources at all. It's important to mention this.
Agreed.
Because I have some competitors in other universities who have loaded the system with all kinds of patents and claims of devices that when I test those things in my lab, they don't do anything. I have tried to maintain my independence all throughout these 40 years of work. So maybe in the future, but not now. One thing I do, people, for methylene blue, I am recommending your group. I say, well, go ahead. And there are people there who know what they're doing, because many people ask, where can I use this or that? And I'm not a practitioner. So you who work with practitioners are the ones that should be leading the way. These devices are coming. I have already seen the prototypes behind me. I have early ones were actually made by my own sons, my two engineering sons, one mechanical, the other electronic engineer. They just help me out in the lab. They work on robotics. I can tell you there's a company that are originally based in London that had a subsidiary in the U.S. that went through the process of the FDA that took several years to approve a product for a condition called aging-related macular degeneration. And this device is already FDA approved for that condition, and is the one and only device or intervention that is being found to stop and prevent neurodegeneration. Nothing else that the FDA has approved has done that. They have refined this and made it in a way I only consulted with them. I have nothing to do with their financial. And I'm so happy that they got this. Is opening the way for some of these non-traditional interventions to be part of the mainstream now. When we did that experimentally with animals, with the ganglion cells, with the mitochondrial toxin, there was a model of what is called Leber's optic neuropathy, which is the most common cause of blindness in young people. And I try to have companies do this for years and no takers, either with the photobiomodulation or directly with methylene blue injected in the eye, which we have done in these animal models and demonstrate its effectiveness.
Okay. The surgery or the process or the treatment for AMD is it's a process. And after the surgery, you have to like lay down, face down. You can't like stand up. You have to have like this air bubble that allows your eye to heal over like 2 or 3 weeks. Like anything that probably avoids that potentially with more light-based treatment or laser-based treatment would be amazing. I assume you said it was LED involved, or is it some kind of combination of?
Yeah, it's LED involved, primarily the red and near infrared.
Okay. And can you like, considering it's goggles for your purposes, can you like just move them up to the forehead to get the correct location that you're after for?
Well, no. For the eyes, there's no problem for penetration.
Yes. Okay. So you need something stronger to go through. Okay.
If anything, your eyes converge the light and make it more intense by the optics of the eye.
Okay.
So
that's fascinating.
These parameters that are working here are not going to work transcranially. Is that now there is a concern by some people that, for example, methylene blue at the low doses that I recommend that you use will have a negative impact on the microbiota.
I've seen some of that.
There is no such a thing. There's only one controlled experimental study in animals that shows a whole range of doses and no effects on the microbiota. So this is a concern that people shouldn't have. These low doses of methylene blue will not affect their microbiota in any significant degree. If anything, they will have a beneficial effect. For example, if you have a urinary tract infection where there's a lot of a particular type of bacteria, when it concentrates, builds up inside the bladder, then it can have this pro-oxidant effect. It was used in the US for decades for those purposes, and then it was replaced entirely by antibiotics. Well, as you know, the antibiotics have a negative effect on mitochondria. You are killing the bacteria, but in a way that also affects mitochondria. Pay attention to the metabolic side of the equation. And the same thing with psychiatric medications. They focus only the neurotransmission, but some of them actually affect mitochondrial function, and others actually help mitochondrial function. But there is right now no medically guided opinion on when to use one or the other. If patients have cognitive decline, you don't want to give them a medication that is going to affect their mitochondrial function. Right now, these things are not connected. So some of your physicians there should start promoting these connections, I believe. I'll try to do my best. In the bipolar study, we revealed one of those connections with one of these psychiatric drugs that was synergistic with our photobiomodulation because it's known to independently facilitate mitochondrial function. If they're going to get these psychiatric drugs anyway, you may as well get the ones that are going to work together with these other interventions.
Yeah, that's a great point. We are aware of like some of the UTI or urinary tract infection implications because there's antibacterial, antiviral impact of methylene blue. That's kind of a cool side benefit, if you will. And I'm aware of some, recommendations. If you're looking to avoid antibiotics, there's some publications out there. This is not medical advice, but there's some publications out there that could present methylene blue as an alternative in some cases. And antibiotics for sure.
Not even an alternative. I don't have any problem having the two together. The advantage is that once you get better.
Oh, like in combination. Oh.
You're not going to have this cycle. That is the problem with the older people, especially older women, that, they get the infection taken care of by a cycle of antibiotics, but then it comes back and they have to go to another one and they get weaker. So having this other compound that it will be synergistic with attacking this infection, it just makes sense. And this may then prevent the next cycle from happening.
Blowing my mind, Dr. Gonzalez-Lima, because I would have not guessed. I kind of saw it as like A or B, but not the synergist combination of the two.
The last word I will say, because my students will appreciate these, if there are people that want to participate in our work, whether it's autism, bipolar, ADHD, or cognitive aging, mild cognitive impairment, all of those studies are going on. People are invited if they are in the area, in the region of Austin. And in many of these, we will be able to give them the device that they can take home when we built enough of these LEDs to start giving them. Hopefully by that time, my trainee will have another source added to the one that is existing now, so that people, if they see a benefit, they can go now into that market. We have faced an ethical problem with that in our pilot studies, like with autism, the parents have seen these benefits and they don't want to give the device back to us. They don't want to keep it. So we have been investing time and effort now on having more of these things that will be available so that people can continue.
Much appreciated.
Thank you so much.
Thank you, Dr. Gonzalez-Lima. This has been compelling, informative, and I would say really just true breath of fresh air. So we sincerely appreciate your time. With that said then, that's it for this episode of Beyond Healthspan. We hope that you are all walking away, and I'm sure you are, I know I am, with new insights, maybe a little bit of inspiration for your own health and kind of healthspan journey to live better, longer, and without the guesswork. So if this did spark something within you, share it with a friend, drop us a comment, or let us know what you want to explore next, or share the good word, because I think there was a lot of great stuff here today that more people should certainly have access to. We're building a community here together that's curious about living longer and better, and we are certainly glad that you all are here. Thank you.
Bye.